So tonight, I'm going to tell you a bit about what the tumour microenvironment is, because this is what I research. I'm going to tell you about some of the important components in it, how we study it, and there I will show you some of our own actually unpublished work, and why it is so important for cancer research and treatment. Well, at least why I think it's so important for cancer research and treatment. And everyone in my lab, I hasten to add. Okay, so this is a tumour microenvironment. So for those of you who are biologists and know about histology, this may make a lot of sense. For those of you who aren't, don't worry, I'll try and illustrate it for you. So on this screen are thousands and thousands of cells. And here, this very deep purple area is actually the malignant cells. But even amongst the malignant cells, you can see some little blue dots that look different from the big malignant cells. Most malignant cells have got quite their nucleus. You know, I'm sure you all know the nucleus, the sort of where the DNA is, the cell, the controlling bit part of the cell. The nucleuses are often quite pale and wide and what we call a rather open structure, whereas these cells have got very dense nuclei. So these are immune cells. And then we've got some very long, thin cells, the fibroblasts, the builder cells of your body. Now, these are red blood cells, so here is a blood vessel. And then this kind of sort of dense area is the extracellular matrix. This is what we all have. This is the stuff that keeps us standing up, and so we're not a jelly-like blob on the floor. It's sort of the matrix that holds our body together. And down here is what was this tissue before the cancer invaded. And this is called the omentum, which is a fatty bit of your peritoneum, but not something we really want to talk about this time of night. But all of this is really the cancer, but only this is the malignant cell. The rest of it has been built, has been recruited, but these are not innocent bystanders. Some of them actually may be trying to attack that cancer, but most of them, in the form you see it here, are helping the cancer grow and spread. To just emphasize that point is another similar section. By the way, if you don't know how histopathologists, the people who study tissues, how they study tissues, or how we do in the lab, it's a very, very thin section of a tissue that's fixed in various things, maybe formalin, embedded in paraffin, and then the tissue is stained with things like food dyes. They're not food dyes, but like you might use to color the icing on your cupcakes. So here I have just blocked out most of the cancer. And can you see how much, again, of that is not malignant cells? So what are the most important components? First is the immune cells, and we're going to focus a lot on the immune cells. So there are many, many different types, maybe over 50. We don't understand how many there are yet. There are fibroblasts, those builder cells, and the extracellular matrix. There are blood vessels. The cancer cannot grow. How do you think a cancer grows? Your organs can't survive without blood vessels. The cancer cannot grow without blood vessels. And then there are a whole host of other things like cytokines, chemokines, all kinds of soluble mediators that allow the communication between all these different cells of the tumour microenvironment. And every cancer, I don't know of one example. Some are what we call very cold. They have very few immune cells, but every cancer contains some immune cells. And here is an example of this. So here, oh, I spoiled that. Here is I think, a prostate cancer and a breast cancer. I've sliced through them. And then using a technique called immunohistochemistry, you don't need to know what it is, just call it IHC. It's a way of very specifically highlighting the immune cells. And if we do that in these two cancers, just again, look how many immune cells are knocking around in that cancer. but they tend to cluster around the malignant cells. Now here is an example of some T cells. T cells are a type of lymphocyte, and this is an ovarian cancer. And here, remember I told you about these very open nuclei. You can see they look quite different, not so dark. These are the cancer cells, this bit here. Then look at all these T cells. They look like they're trying to get to the cancer, but they don't. There's very few in amongst the cancer cells. Here's some macrophages. This is another type of ovarian cancer. This is called clear cell ovarian cancer. And this is one of my clinical fellows, MJ. And I told you some T cells, but here we've got macrophages. And again, macrophages sometimes can come in closer to the malignant cells. But this is from a patient, and there's plenty of immune cells here. This little area of ovarian cancer, again, the macrophages, we've highlighted them in brown. That's very specifically picking up macrophages. And you can see in this case, they're sort of weaving around the cancer cells. Then here's an example of some fibroblasts. So these are the long, thin cells that tend to wall off the cancers. Cancers also need blood vessels. And the angiogenesis, as we call it, the new blood vessels in a cancer, is chaotic. It's not organized like the vasculature of the rest of your body. And here's an example from one of my colleagues in the US. So in this, again, a tissue section from a cancer, the blood vessels are highlighted in blue. So you can see some blood vessels here. And in green, that shows the cells that are able to really properly metabolize. They've got enough oxygen. But not much more than 10 cells away, these cells here stained red are what we call hypoxic. They haven't got enough oxygen, so most of them die. But some of them will mutate, and they become even more dangerous because they can survive on very limited oxygen, so they can survive in really the worst conditions possible. And this is, again, a very bad sign if a tumor is too hypoxic. I talked about the matrix. So here we stained the matrix up. This is collagen. And look at this tumor. Look at the architecture of this collagen in the tumor. So in pink are all the cells, cancer cells, immune cells, but amongst them is this collagen. And my friend and colleague, Vicky Sanz Moreno, we just recruited her as another professor to Bart's Cancer Institute. Vicky showed some amazing images the other night of cells moving in extracellular matrix, and she very kindly let me borrow them today. So can you see here how cells use the extracellular matrix to move, but also it can contain them. And if you look here, any engineers in the audience might understand it more. Those arrows show the vectors, she tells me. I don't know, I think this is a melanoma cell. And here the different colors show the different stresses of all the matrix. But I also wanted to give you an idea that this is not a static thing. You know, this is a living, vibrant, moving, very dynamic microenvironment. And the way in my lab we've recently been able to do this, which at the moment I would call pretty science, but I think we're going to be able to use it to preclinically test some of these new treatments I'm going to tell you about, is that we're able to take a slice of a tumor from a patient, make a slice about 1/3 of a millimeter, embed it in agar, and then put it under a special microscope. And for about a day, we can keep that alive like it is in the body. So I'm going to show you what that's like. And if I can run the movie, I've lost my, hang on, I need to put my glasses on, I think. seem to have lost, here we are. So can you see here, I'll run it again. Before I run it again, I'll tell you. So green here, these are the malignant cells, and these pink cells and red cells are different type of immune cells, and the blue here is the extracellular matrix. And if you look, you can see how, oh, actually it's moving quite slowly. I think maybe we've got a power problem here. That's a bit annoying. Let's try the next one. Oh, you can see, I think. And then this one is particularly interesting if it works. Yes, can you see here? So here, the tumour is stained in red. This is called confocal microscopy, but spinning disk, actually. And here is the extracellular matrix. But here, you see the immune cells are green and red, but we don't have many of them. And this may just be anecdote. This is not, we haven't proved this in any way, but what we're fascinated about is normally the malignant cells stay quite still. But if you look here, some of these malignant cells are actually moving, and it looks as if they might be spreading out. If you look here, we've never seen this before. But it's interesting that it's happening where in an area of the tumour where there aren't many immune cells. So we can look in all kinds of different ways. I'm just giving you some examples. We can study this tumour microenvironment. And I've showed you examples of many different common cancers, but they all have these immune cells. So how do we study the tumour microenvironment? I've showed you some examples. What's really important to us is that the research should be led by what happens in the human. We need to study the real human cancers. There's been, a lot of time in cancer research where people have worked a lot on mouse models. One of the mottos of my lab is if you're a human being with a mouse cancer, we can help you, But we passionately believe now we have the technologies, like some of those things I've shown you, to study human cells and human cancers. Mouse models are necessary, but also what we call in silico. We have big data now. We have vast amounts of data on many different types of cancer, and it's a wonderful thing. It's freely available. It's fantastic. The only snag is we need very clever bioinformaticians, mathematicians, to really be able, computer programmers, to really be able to help us interpret. But there's a vast amount of big data. Oh, and we need big computers as well. But also, and this is what I'm going to tell you about as well, is that it's an interest of mine, it's becoming much more used, is that because we do want to be led by the human cancer, we're beginning to apply some of the technology that's used in tissue engineering, say to try and engineer a patch to repair a failing heart or an artificial kidney or bladder. We're using some of this technology to begin to model cancers, not in mice, but ex vivo in the laboratory, but this complex tumor microenvironment. So I'm now going to tell you a little bit about some of our own research, and I hope you will understand. So we work on a type of ovarian cancer called high-grade serous ovarian cancer. And five years or so, I had some funding from the European Research Council to build a human tumor. Now, some of my colleagues said, well, we've got enough cancer, why do you want to make some more? But obviously, there were good, I hope, scientific reasons. But instead of just trying to model this outside the body, we first decided to deconstruct. And so we took samples of the cancer when it has spread to this tissue called the omentum, and on each sample, we did various different experiments. We measured its stiffness. So for those engineers in the audience, we measured the tissue modulus. Now, this is important because doctors know that cancers are generally stiffer than the surrounding tissue. And we now know that this stiffness actually is another stimulus to the cancer. It encourages cancers to grow and spread. We looked at that architecture, how much of the tissue was remodeled. We looked at 8 different cell types. There's many more, but that's all we could afford, to be honest. We looked at the load of those chemokines, cytokines that I told you about. Then, don't worry about that term, but we looked in great detail, more than anyone had ever done before, I think, at this extracellular matrix. That's all the components. And then here we looked at the way the genes were being transcribed. This is an idea of what proteins it was being made by the tissue. So we ended up on just 36 samples, but we ended up with what the younger generation might call TMI, too much information. And this was published in a journal called Cancer Discovery this year. So we ended up with the tissue modulus, which was a simple score, but then for each sample, we ended up with all this information. But thanks to the bioinformaticist, it began to make some sense. And I'm not going to tell you anything else, just I'm going to tell you one thing that was unexpected. We discovered a lot from doing this. We discovered, if you like, a template for how we might try and build this human tumor, and a way of validating it because we had all this information. So that was exciting. But then we found something quite unexpected because, as I told you, nobody had really measured very systematically the stiffness of tumors before, and nobody had looked at the way this matrix is remodeled in such detail. And what we found was that there was a particular pattern. You don't need to know how we found it, but there's a particular pattern of that matrix that actually was a very bad sign. If that pattern was there in a cancer, then the patients with ovarian cancer would generally not respond so well. You know, they wouldn't live so long. It was what we call a poor prognostic sign. But just out of curiosity, and because we've got some very helpful bioinformaticists, we went to that big data, we went to the big computers, because we found this in ovarian cancer. We wondered whether we find it in other cancers. And basically we did. We call it the matrix index. Again, that doesn't matter. But looking at the big data, we found patients who had high level of this pattern of matrix and compared them with low. And this is the survival over years. And then we looked in lung cancer. And then we looked in kidney cancer. And then we looked in liver cancer. And we went on and we found all in all that 13 different cancers, we could use this pattern, we'd have to look at RNA, we could use this pattern to detect right at the beginning that those patients would do worse. Now, the reason I'm showing you that, we don't understand, incidentally, why. And now I've got 5 years research funding, and I've got other people and other groups are looking at this, and it's become a hot area, not just from our research, but for others. But the reason I'm showing you this is that a lot of cancer research these days subdivides the cancers into ever decreasing, which is important. It is very important. But there's some things in the tumor microenvironment that are common, and it made us wonder. These matrix proteins are ancient proteins. They're conserved throughout evolution. Maybe the body's response in terms of the matrix is something that is common to many cancers. Now, this is just it's just an idea at the moment, but if it is, then there could be partially the way that we might in the future treat cancer is something that will stop, that will prevent some of this matrix reaction. So that is for me, a very exciting area, and I have funding from Cancer Research UK now to pursue that over the next 5 years. And I'm very excited and happy about that. But we can't just, how can we get at this? Is this true? Can we target it? You know, if it is, it would be really exciting because it could mean that this is something we could add into a treatment regime for many different patients. So we do need to have some mouse models. So we made some new mouse models. But of course, the great thing was that we already knew a lot about the human because we developed all this too much information on the human cancer. So we made some new mouse models, and we were careful to make the models so that the original genetic mutations found in the human were also found in the mouse. Now I'm going to show you a heat map now. So bottom line here is that our mouse models, because we made them carefully in the beginning, have many similarities to the human. So a heat map, this is called a heat map because blue, there isn't much of a particular RNA molecule. And remember, RNA goes to protein, so this tells you really essentially what proteins these tumors will make. And red means there's a lot of it. This is very easy to understand, I hope. Each one of these columns going down is an individual sample. All the blue ones are the normal tissue from the human and from the mouse. And you can see they're all blue. And then going across here, each one of these columns is either a human tumor or a mouse tumor. And you can see that there's a lot in common. And so here we think we have mouse models because we studied them carefully that will be useful in our experiments. And what else we can do with all this big data that we've got is the immune cells leave signatures hidden in the big data, hidden in this RNA. And each of these colors represents the signature of an immune cell. And you don't need to understand that. All you need to do is look at the pattern here. And then we can look across. This is our human, and these are our mouse models. And you can see that they have a similar pattern. So that means that we can use them for the human, to research the human disease. And then finally, the matrix that we found, that pattern, this is the human normal, this is the human diseased, and the levels in some of the mouse models and the tissue modulus, here's the tissue modulus in the human and the mouse, is the same. That means that we think we can begin to try and understand how that matrix makes cancers worse. Our hypothesis is that it blocks, it sticks the immune cells, they can't get to the cancer because we can model it in these particular mouse. And here, this is a very clever way of looking at 6 of these most important matrix molecules. If there's just one, and this is the normal tissue in the mouse, it's blue. If there's 6, it'll be red. And you can see as this one is sort of quite pale, but this one, which is this one with the really highest, well, one of the highest tissue modulus, is very, very red. So it's very stiff. And so what this gives us is the ability to begin to understand and experiment and find out if we can. So we've got some mouse models of the tumor microenvironment, and that's what we'll be working on. But if you remember what I told you a while ago, we had a grant to build a human tumor. Now this is completely crazy. This is really a lot more difficult. So how far have we got? So going back to what I showed you before, this is an ovarian cancer with lots of immune cells, but loads of fibroblast and matrix, and this is the original omentum, which is made mainly of fat cells called adipocytes. So we decided to try and make an artificial omentum. So we get omentum from the patients, and basically we grow adipocyte gels, and we also grow malignant cells and fibroblasts, and then we put the two together. So we call this our fat farm. And it works. So little green here, that shows the cells are alive. And then what we end up with is something that really does, don't worry about all these signals. Again, we're identifying the brown here is the stain for the malignant cells. These are stains for fibroblasts. This is one of the cytokines. These are the matrix molecules. And this is what it looks like just through a section. And we're beginning to trick the pathologist by saying, what do you think this is? And then sometimes they say, oh, yeah, you know, that's a mental metastasis of high-grade serous. And we say, no, we grew it in the lab, which is quite exciting. And here's a nice diagram of what we also do, which is that was putting the tumor cells in, but we also actually create an artificial omentum with the fat cells, with the fibroblasts, with some lining cells called mesothelial cells, and then we put the malignant cells on top. And basically, you can see here that, again, green shows the cells are all alive. And then this is the actual human tissue, and this is the mouse model. So, sorry, the human model. It's not mouse, it's human. And this is the brown that detects the normal layer. So we're beginning to be able to create parts of the human tissue. And in here, we've got the matrix. The tumor cells are in green and white, and the matrix is all laid down here. And this is really very similar, and we were able to make RNA from these, and we found all the molecules that we found in the human tumor in these models. Now, of course, we haven't got the immune cells in there yet. That's what we're working on now. But it gives us another way of trying to understand without going into the mouse models, which are expensive, time-consuming, and we can look at real human cells. But also, I told you about the blood vessels. So here we come to more bioengineering and microfluidics. And so this is, microfluidics means tiny bits of fluid flowing through some engineered matrices. And here are some blood vessels that we developed. And here, they're blood vessels. Can you see they're growing towards something? And they're actually growing towards a spheroid of tumor cells. And Joash Joy, a PhD student in the lab, is working with, this is his model. This is his little plan for his microfluidic device. There'll be fluid going through here, and he'll have one of our little adipocyte cultures in here, put the blood vessel cells here, and then he'll start delivering immune cells. And that's a collaboration with GlaxoSmithKline, and he's going off soon to GSK to make some immune cells, genetically engineer some immune cells. And this, if we get the movie working, this is what it looks like in reality, how the blood vessels are growing into that microfluidic area. So we have the blood vessel cells at the top, and then you can see, and they really do develop what we call lumens. We can even put supporting cells called pericytes over that. Then also the idea is we may be able to study cancer cells spread into blood vessels by putting cancer cells in there. And so our ultimate aim, of course, is to have cancer on a chip. So why do we use the word chip? Because these microfluidic devices are, I don't quite know where the term came from, but the microfluidic devices are about the size of one of those USB drives. And I'm now going to show you a little movie because we're about to start to try and develop this cancer on a chip. We're about to start a collaboration with a company from America, from Boston, that's a spin-out from an amazing bioengineering institute called the Wyss Institute, and it's a company called Emulate, and we're just going to start collaborating with them to build a human tumor microenvironment on a chip. And they have lent me a video that I will show you with sound. I hope it's going to work.
The alveolar lung chip is crystal clear, flexible, and about the size of a AA battery. Its two central channels are separated by a porous, flexible membrane. The top channel is lined with human alveolar epithelial cells. The bottom channel is lined with alveolar capillary endothelial cells, faithfully recreating the alveolar capillary interface of the human alveola. The epithelial surface of the top channel is exposed to air, while blood, or a medium containing human white blood cells, flows through the bottom channel. By mechanically stretching and relaxing the chip, we can recreate the natural forces our cells experience when we breathe. The natural immune response of the human body can be recreated in the chip by introducing bacteria into the top channel and white blood cells into the bottom channel. As white blood cells flow through the bottom channel, they respond to signals from the epithelium and are drawn across the membrane to engulf bacteria on the other side. The alveolar lung chip allows us to recreate both healthy and diseased states of the lung, and provides a real-time window into the inner workings of human biology. It is one part of the human emulation system, which is allowing us to gain a deeper understanding of human biology and health.
And it's just that big. So my university has just entered into an agreement, and we've got a whole Emulate Centre, and we're going to get, it's lovely, the machines that the chips go into are called Zoes, after the Greek for life, and then there's something called an orb that actually powers it and everything. But it's absolutely fascinating, because that lung chip, that the cells, you don't get anything like the same reproduction that you would expect unless you actually subject them to those forces. And so we're going to work with them. Now, because we work on peritoneal cancer, we need peristalsis, the movement of the gut. And so we will be taking what we've got so far of our tumor models into those chips, but with the peristalsis motion of the gut. So hugely exciting. But, you know, it's not just for fun, this. I think this really is a way of looking at new treatments that attack that tumor microenvironment. So, I've told you a little bit about the tumor microenvironment. Why is it important to study it? Well, these non-malignant cells in a cancer, they're not innocent bystanders. We need to understand the good and the harm that they do. And many new cancer treatments are based on tumor microenvironment research. So I'm just going to summarize and show you how the treatments develop. Don't worry about the sort of technical term here. It's a lovely diagram done by one of my recent PhD students. But if you imagine, here we have a cancer cell. And as it begins to turn more malignant, it makes those cytokines and chemokines that attracts all these and many more cells to the cancer to help it grow and spread. Now, as this happens, it's a sort of chronic inflammation. We were talking over supper, inflammaging. It's a low-level chronic inflammation that fails to reverse. What got me into this field was a very long time ago, a wonderful review from a guy called Howard Dvorak in the New England Journal of Medicine that was cancers are wounds that do not heal. And there are many analogies between wound healing and cancer, except the cancer, the inflammation doesn't resolve. These immune system, these immune cells, there's T cells that are capable of killing the cancer, but they're what we call, they're exhausted. they're inhibited, they're suppressed. The tumour is stiffer, I've told you that. The increased stiffness adds to this positive feedback. There's lots of new blood vessels, but there's also lack of oxygen, which stimulates mutation. But at the same time, new antigens, the tumour is different, and the immune system ought to be able to recognize it. And so there's a whole load of new treatments that come from this understanding of this tumour microenvironment. And that's my student who drew this. So coming back to those T cells, remember those T cells? There they are. There's loads of them. Now, a lot of people used to think they were innocent bystanders. They were just there passing through, you know, just part of the rubbish that's cancer. They're not going to be any good. But it's probably about 10 years ago now. Where are we? A little bit more. There was a paper published in the journal Science where they'd studied just on simple tissue sections like I showed you, just like that one. They'd studied the number of T cells. They'd actually counted them. Now we have fantastic digital high-tech solutions for doing this. We don't sit there going 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. We have a computer that sorts out the brown from the blue cells and saves us a great deal of time. But they counted the number of T cells at the margin. They counted the number of T cells in amongst the cancers. And they found that the more T cells a cancer had when the patient was diagnosed, whatever the stage, the patient would do better. Now here's an example from ovarian cancer. And you see the first paper on ovarian cancer was 2003. And if a patient didn't have any T cells in their tumour, that's their survival. But if they had a lot of T cells in the tumour areas, they would do a lot better. And then a later meta-analysis looking at lots of patients, and this is now 1,800 patients. You see, if you understand statistics, we have a highly significant p-value. Now this really was the first bit of information for years and years, because people had tried all kinds of immunotherapies, never anything really convincing. But this doesn't really prove T cells, but there's a very strong association between the number of T cells that a cancer has and the way the patient, you know, how long the patient will live. And this is true for many different types of cancer. It's another of those things that are common across tumor microenvironments. So, how do we change that tumor microenvironment? How do we wake up those immune cells? We have vast armies of T cells, of macrophages. We have this matrix that maybe is trapping them. How do we change that microenvironment and improve survival of patients? Well, the most famous, and this has been now it's 10 years or more, is what's called immune checkpoint blockade. And I'll tell you more about that in a minute and give you some data. But basically, our immune system is incredibly powerful. And when you have a cold or a flu, you get better, don't you? This is because your immune system, it doesn't matter if you take aspirin, paracetamol, you're still going to, whatever it is, the course of the disease is going to be the same. You might feel a bit better, but you're not actually helping. It's your immune system that fights it. But your immune system doesn't want to go into overdrive. It needs to shut off. And so there are these molecules that were discovered by Jim Allison and Honjo, I forget his first name, in around 1993, 1996. They got the Nobel Prize last year. And these stop your immune system overreacting. But what happens is the cancer corrupts it, and the cancer switches this on. So you can have those good T cells sitting in the cancer, but those immune checkpoint molecules are there, and it stops the T cells attacking the cancer. And what this treatment does is that it blocks those immune checkpoints, and it wakes up the immune system. And in some cases, some of the time, you get quite an amazing response even to metastatic cancer. The other big advance really comes from those T cells that I've talked about. Either by taking T cells, taking a biopsy of cancer, extracting the T cells from it, which can be done quite easily, and then stimulating them with cytokines. And so you can grow up 100, 1,000 million of them, and you put them back into the cancer in the patient's own T cells. So you've grown them up, you've woken them up, and sometimes you can get a response. But also these CAR T cells, so in this case, the patient's T cells are taken from the tumor, and then they're genetically engineered to home back to the tumor. And this has been particularly effective in children who have leukemia, because most children can be cured of their leukemia, but these are children whose leukemia does not respond or has relapsed, and the CAR T cell therapy works there. So we know that these immunotherapies now work. Also, there are drugs, antibodies, and small molecule drugs that attack the blood vessels. We really thought this would be fantastic because it seems so simple. You knock out the blood vessels, the cancer's going to die because it hasn't got enough oxygen, it hasn't got enough nutrients. But the trouble is, the cancer produces many factors that stimulate blood vessels, and what happens is you knock out one of those factors with the drug, and the cancer just makes another one. And so what happens is that most of these, what they do is they will have, the patients will have, they will not relapse for a longer period, but when they do relapse, it doesn't seem to affect overall survival. It's not true in all, and there's a big proviso in all of this that I'll come to in a minute. There is a couple of vaccines now. Of course, the most important vaccine really is against HPV and in preventing cervical cancer. So now all young girls, and I hope soon, I'm not expert on this, maybe young boys, because and this should eradicate cervical cancer because the HPV virus is necessary and sufficient for cervical cancer. But also what I think is the future, and I think is increasingly exciting, is not only do the malignant cells make the immune cells have this immune checkpoint molecules, but also they have a signal on them called "don't eat me." Again, it's part of the body's natural physiologic processes, but the malignant cells use it so the immune cells can't recognize it. And an antibody to those, that "don't eat me" signal, wakes up those macrophages. But also, there are some molecules that are being tested in the clinic that stop those bad macrophages. I'm more interested in sort of waking up the macrophage army, because there are millions and millions and millions of these macrophages in the tumor. And I think it's going to be very exciting if we can find ways to switch them, re-educate them, wake up and smell the coffee, destroy that cancer, and then finally break down the malignant matrix. So these are all treatments that are now in routine use, and these are the ones that are coming along. And I think this is sort of obviously an exciting area for me. And then finally, we have all this fascinating data that people who've taken aspirin for long periods of time have a decreased risk of cancer, and also if they get cancer, it spreads less. But this data is very controversial. You may have read in the newspapers, people over 70 shouldn't take aspirin, it's not doing any good. And there's, you know, it's all big analyses of hundreds of thousands of patients who've been studied for long periods of time. We need to understand it, but it's very intriguing because obviously if we can find drugs that will sort of attack the tumour microenvironment of the very, very, very earliest cancers, this would be the most exciting thing of all. So if we think of where we are, chemotherapy, just a hypothetical cancer, over time, this is what chemotherapy will generally do. I mean, this is a hypothetical. I'm not talking about one cancer or another. I'm just talking about in general. Then, because of all the genetic research that went on, there's been a whole host of very exciting new drugs, which we call targeted therapy, which are meant to just attack the cancer cells because they attack those oncogenic pathways. They were great. Some really dramatic responses, but often the cancer comes back. And the way, it's just like those anti-angiogenic drugs, the cancer finds increasingly complex and convoluted ways of escaping. It mutates and escapes the targeted therapies. And then along came, in some cancers, some of the time, the immunotherapy. And what was striking about this was this shoulder. It's never been seen before. There was that 20% of patients who were alive that shouldn't have been alive. Now, we still don't have a clear idea of how to detect those patients who will respond. But what's happening now, and this is primarily, and we must remember this in malignant melanoma, is that there's two different ways of blockading those immune checkpoints. And what you see is the curves are coming about there, maybe about there. So this could be weeks, this could be months, this could be years, depending on the type of cancer. But what we're all thinking now is that if we can combine different types of immunotherapy, or the targeted therapy with the immunotherapy, or even the chemotherapy. A lot of the research in my lab shows that chemotherapy, you'd think chemotherapy would destroy the immune system, but what it does is it creates a load of damage. It releases new antigens, and actually all the evidence from our studies and others is that chemotherapy can actually wake up the immune system. So maybe in cancers where chemotherapy works, we want to give the chemotherapy and then we come in with the immunotherapy. So a little update from my clinical fellow, MJ, who did this for me, so that I wanted to, because I'm not a clinician. So where is the immune checkpoint blockade being used? Well, interestingly, there are some human cancers that are driven by viruses. And these is where they're seeing very, very interesting responses to immune checkpoint blockade. This one in particular, it's a highly lethal skin cancer that's caused by it's a very rare disease. And in some patients, there are dramatic responses. Cancers with high numbers of mutations. I think there's a clue here. These are quite antigenic. They're easily recognized by the immune system. Some types of colon cancer. But also, what is happening for most cancers in most clinical trials now is that there's combination with chemotherapy, with targeted therapy, other types of immunotherapy. And there's a lot of trials going on in this area at the moment. They tend to be a little pragmatic. There's not that much, I don't think, scientific rationale, but I'm hoping that the models like we have and others are developing, the human models, the more sophisticated mouse models, where we think that they have a tumour microenvironment that resembles the human more. Our hope is that we can develop more rational ideas. And it's particularly in ovarian cancer, which we work with, because the patients respond very well to initial chemotherapy, but many will then relapse, and it's a law of diminishing returns. They're increasingly resistant. So a lot of our research in the mouse models will be to give the mice chemotherapy, and we find that mimics what happens in the human patients. We can study it in the humans and the mice, and then try tumour microenvironment therapies to see how long we can stop those mice relapsing. And from that, I hope we'll be able to translate back to the humans and, you know, in a few years' time, have a clinical trial. So, is immunotherapy the answer? We've learned about the tumour microenvironment. The most powerful cells in that tumour microenvironment are the immune cells. Immunotherapy answers. You know, immunotherapy harnesses our amazing immune system. Well, only the immune system has the power and flexibility to deal with the moving targets of malignant cell clones. Cancer is evolving in the body the whole time. So only the immune system could cope with that complexity. And the successful, and I must stress this, it's a minority of patients with a minority of cancers that respond to these immunotherapies. But I think it is a light at the end of the tunnel if we can just enhance this. And what's really exciting is when they work, that a patient can have metastases around the body and they all respond, which shows us that we can really wake up the immune system. So this is all very positive. And also, although many people have a reductionist approach at the moment of subtyping cancers and subtyping metastases, but I think things like the T cells, the matrix response to cancer, they go across many cancers. It could be that when somebody's diagnosed with cancer, and I've heard some some of the top, you know, people I most admire in terms of clinical medical oncologists say this, that a patient will not be, that patient's got breast cancer, that patient's got kidney cancer. They will look at the cancer and they'll be able to say, well, that patient's going to respond to immunotherapy, so we refer them to the immunotherapy clinic. Or that patient won't respond to immunotherapy because their tumour is cold. It's just suppressed the immune response. So first thing we've got to do is give them targeted therapy, chemotherapy, matrix therapy, something to let those immune cells in, and then we'll come in with the immunotherapy. Now, this may be the way that things will go in the future. So there is a potential to move away from this reductionist approach. It is, you know, it's very, very important because in some of the top cancer centers, they will type the tumor for all its mutations, and then for when it stops responding to one targeted therapy, they will say, well, then we can use a BRAF inhibitor, or we can use a MEK inhibitor, or whatever. And this is really, really important, and it may work. But the alternative approach is to, which excites me more, which is to sort of really harness the body's own defenses and wake up the immune system and allow it to do the job that somehow, as the cancer has evolved, it's failed to do. So there's experimental evidence for immune surveillance. I haven't gone into it, but there's some quite good experiments from mouse models that there is what we call immune surveillance, that the immune system is continually surveying our bodies for malignant cells, and most of the time successfully. We know we can use vaccines for cervical cancer, and I've talked about cancer prevention. So, but the proviso is that many patients and cancer types do not respond to current immunotherapies. There's some evidence that some cancers may find ways to escape immune control. So a lot of people now think that we need to target different components, those different components of tumor microenvironment that I told you at the beginning of my talk. The immune cells, the fibroblasts, the cytokines, the chemokines, the blood vessels. Maybe sequentially, I don't think you could do it all together without targeting the patient. And also remembering that some of the older treatments that we've used for cancer may stimulate the immune system. We've got to understand that more and exploit it. So I will stop there, but if anyone would like to remember this lecture a little bit more, I also am very interested in informal science education, and I run a science centre for children down in the East End of London called Centre of the Cell. And we had over 180,000 young people interact with us over the last 10 years, but we also do apps. And so there is an app that you can download on your smartphones, free, called Tumour Takedown. And just this brings back the message. So there's my avatar, by the way. But with this, you can build your tumour microenvironment, and then you can attack it with CAR T cells, with immune checkpoint blockade. You can stimulate some macrophages, and that helps. I hope some of you might download it. So there's a lot of my team and collaborators that I've worked with over the years and working with at the moment, and also acknowledging all my funding, and there's Tumour Takedown again. Thank you.
Well, Professor Balkwill, that was fantastic. Cancer always seemed to be a bit of a black art that nobody wanted to talk about. And gradually, it seems that scientists are getting to the bottom of it. And you've explained, insofar as we're capable of understanding, with great clarity, what appears to be a hugely exciting new opportunity and approach. Now, I know there'll be questions from the audience. Can I suggest we adopt the new model? That is, our lovely assistants at the back there, chose my words ill-advisedly, will keep an eye out for people who'd like to ask questions, and I will look to them to choose people to ask, and then I'll flip from side to side. So would you please try and catch their eye rather than mine, and then that makes my job easy, and they do all the work, which is the objective of this approach, as you'll appreciate. Yes.
Thank you for a fascinating talk. I'd like to ask a kind of a mechanistic question, I suppose. You mentioned at the beginning that tumour tissue is stiffer. I'm an engineer, so it appeals to me. Is it known why it's stiffer? Is the cancer adapting to make itself deliberately stiffer, or is it just an accidental byproduct of the fibroblasts?
Well, I think it's the former. Because if the cancer is stiffer, we mustn't think of the cancer as some evil being, we mustn't anthropomorphize it. But obviously, if it's stiffer, then it evades the immune system because it has more matrix. Also, actually, drugs can't get in. For some cancers like pancreatic, it's so stiff that the drugs can't get in. But also, there's quite a lot of research that stiffness actually, there's some very intriguing research from a few years ago where they created situations where pre-malignant cells were exposed to stiff matrices, and that was enough to turn them into malignant. So it's something that really aids the cancer to grow and spread.
Another
one over there. Oh.
Yes. And what you described in antimicrobial research, if you replace those cancer cells with some tuberculosis cells, what you're describing is caseation.
Yes.
Which we've seen and is extensively researched. And TB, of course, shows the same immune privilege where the T cells fail to get into the caseated nodule. So I just wondered whether you'd gone down that track and looked at some of the TB research. But the other point was I just wondered whether the system responded to anti-fibrolytics to give you more access.
Yeah,
so
that's
kind of two questions.
Yeah, That's exactly, well, just to answer the second question first, which is, would the cancer be likely to respond to antifibrinolytic, drugs that might be used, for instance, for fibrotic lung disease? It's very interesting. I've just started looking at the literature because some cancer drugs are already used for fibrotic lung disease, and there's very much, the body, nature is very economical. There's only a certain number of ways that the body can respond to a noxious stimulus. And I think a cancer is, and I think the fibrotic response, I've got some very preliminary evidence that the lung fibrosis you see in some lung diseases is very similar. And of course, it does lead itself to cancer as well. So I think this is where a lot of research is going. And it's certainly, we've set up a mouse hospital at the moment, where we've got cohorts of mice who we're giving chemotherapy to, and then we're trying to prevent their relapse. We've just started it now. I don't know if it's going to work, but and so one of the things we'll be trying, and in fact we did one experiment and got some very intriguing results that I still don't know whether it's good or bad. So I think that a lot of research will go in that area. The only snag is that if you disrupt the cancer too much, then you could actually make it worse and make it spread. So you may want to just make it a bit softer, get rid of some of that matrix, allow the immune cells in, but don't carry on doing it because that could make it worse. And we've seen this in some of our human tumor models in the lab, that we use a drug that, if you may understand it, that neutralizes TGF-beta, or the receptor, which is, it's a cytokine that's one of the major stimulators of this matrix. And cancers, which normally get quite walled off in our lab cultures, suddenly begin to go all over the place. So it's, and you're absolutely right about tuberculosis, and there's lots of similarities, I think.
I think you may have one more on that side, yes.
That side's winning at the moment.
Hello, thank you very much for that. That's really interesting stuff. I am intrigued by the idea of these malignant cells as an extreme immune response rather than a toxic thing that is then needing an immune response to kill it off. And I don't know whether you've looked into the idea of it being such an extreme immune response that is why the immune system then shuts down. Because you talked earlier about it being one of the genesis for cancer being a wound that doesn't heal, which is something that I've certainly been aware of for some time and makes a lot of sense. And if that is the case, then what's happening is the body is healing that wound in a very extreme way. Thus, the immune system is not getting any messages to react because it's being kind of fixed. And so it's very badly expressed because I'm not a scientist.
No, but that's exactly right. Cancer is a wound that does not heal. And the processes of wound healing are such that they would encourage a cancer. If you see, do you see what I mean? It's a different immune reaction. It's not so much an overreaction. An overreaction would be more like an autoimmune disease. And an overreaction actually is not a bad thing. So what happens with immune checkpoint blockade is you do get an overreaction because you're taking the brakes off the immune system. And so actually the patients also get symptoms of autoimmunity because the borderline between the cancer and the normal tissue is quite narrow, and so the medical oncologists who are used to dealing with the toxicity of chemotherapy are now having to learn how to deal with autoimmune disease. And so it's not so much an overreaction, it's an underreaction. And what this goes back to, don't get me started on this, but very briefly, the only systemic treatment for cancer in the end of the 19th century, beginning of the 20th century, was something called Coley's mixed toxins. And cut a long story short, that was, this guy, William Coley, used to give patients a bacterial broth that would absolutely stimulate their immune system. They get rigors, they get pyrexia, very high fever. And if you read the case histories, he was actually curing cancers that we can hardly touch now. And with the CAR T cells, you get a similar reaction. It's what we call a cytokine storm. It's almost lethal. And that massive overstimulation of the immune system can actually sometimes cure cancers. We can't reproduce it now. It's lost in the midst of time, but it's very, very intriguing. So it's much more an under rather than overreaction.
Left-hand side, going strong.
I don't know if I'll survive the evening at this rate. Thank you. This is a bit of a left field question. I've heard that this is not directly related to your talk, but I've heard it said that we fund female-only cancers far better than we fund male-only cancers. Is this a bad story, or is there some truth behind it?
I think it's a bit of a bad story, because most cancers affect both men and women. You know, yes, there's breast cancer, ovarian cancer, but there's not many other cancers that don't affect, and there's prostate cancer in men, but most cancers do affect both men and women. And so most of cancer research, you know, is about cancer in general. And of course, the kind of cancer research I'm talking about may not be specific for, you know, any cancer or men, women. So I don't think it's true. I don't think it's true.
Could I ask a question then, please? Since there's a little bit of a gap. If you're giving a patient chemotherapy, often it's given in separate courses over the course of months. How do you think you would decide when you finish your chemotherapy before you start your immunotherapy? What's the sort of sequence?
It's a really good question. We obviously, in patients, it's difficult to get many, you know, we can't keep on taking lumps out of the patients while they're having the insult of chemotherapy. So in ovarian cancer, which is a women's cancer, what happens to many of the patients these days is they have 3 or 4 cycles of chemotherapy. Then they have about 3 months rest, and then they have what's called interval debulking surgery. So basically, you give them some chemotherapy to reduce the cancer load, and then you go in and do the surgery, which makes a lot of sense. It's great for us because we have the biopsy before they started the chemo, and then 4 weeks after the 3rd dose of chemo, we can get another biopsy, and we can see how the tumour microenvironment has changed. And we're doing the same in the mice. We're giving the mice 3 doses of chemo. So we know. And that's the only time point we've got. And there's no really good measure in the blood that we found. We know that about 4 weeks after the last dose of these 3 chemotherapies, that there's some evidence that the immune system is activated. So that's probably right. But that's a bit of guesswork, and it's going to be very difficult to be sure.
David.
Thank you. Thank you for your most fascinating paper. I have 2 questions. One is, does the immunization process involve T lymphocytes?
Yes.
And second point is, my interest in the past has been mostly in connection with diet and cancer, which is a major cause. Yeah. Seems to be. Our evidence seems to show that most cancers, the cancers we've looked at, are controlled by inhibiting the absorption of the carcinogen. But how is obesity related to cancer?
Yeah, the question was, how is obesity linked to cancer? As many of you know, there is a strong association with some cancers, in particular in obesity. It's not fully proven, but it comes back again to inflammation, because obesity, with lots of adipose tissue, you've got lots more of those bad inflammatory cytokines, and that's the sort of simple, generally accepted theory. And so in obesity, then the immune system is altered, basically. And so I think that's, as regards carcinogens, well, what we always say about sort of, if you like, inflammation and cancer, you know, mutation, genetic damage is the match that lights the fire, and inflammation is the fuel that feeds the flames. And that also comes with obesity. And I think, you know, that this is a very exciting area. For instance, there was a clinical trial that really excited me and my colleagues who work in this area that was published year before last, where this brilliant cardiologist in America, a guy called Paul Ridker, had this hypothesis that an inflammatory cytokine called IL-1 would be a major cause of heart disease in people who'd already had a heart attack, were smokers, and were generally, you know, very poor and often obese and had high inflammatory markers. And so he did this clinical trial on 10,000 patients, and he reduced their risk of getting another heart attack. But what was absolutely fascinating was he also reduced their risk of lung cancer.
My point was, I should have made it, is vaccination related to T lymphocytes?
Is vaccination related to T lymphocytes? Yes, yes. I mean, vaccination will stimulate the adaptive immune system, which is T lymphocytes. And so, but vaccination probably in most cancers is not going to work once a cancer is developed. Where vaccination is probably going to be most effective is in things like cervical cancer, where the HPV virus, you know, is sexually transmitted, and if you can prevent that infection in early adulthood, then you can prevent the cancer developing later.
Thank you. And the right-hand side here.
Good. Oh, well done, the right-hand side.
Do something for the team. I got two questions about the things you mentioned about the matrix. One was that the stiffening by itself causes the biological response. So that's the first question. Why would physical change cause a biological response? And the second is, you said it's a very ancient observation. It's a very ancient result that get this. So why is it preserved?
Right, to answer your first question, why would stiffening have a biological... If you remember that wonderful movie from Vicky Sands-Moreira, where there are receptors on the surface of cells called integrins that interact with the matrix. And when they bind to the matrix, it sends a whole load of biological signals, massive change of biological signals in the cell. Each cell also has a skeleton called the cytoskeleton, and when it binds to the matrix, the cytoskeleton changes, and these cause profound biological changes in the cell. You take a cell out of its normal matrix, and it goes haywire, you know. So that's, and as regards what's the ancient response, what I meant was that these matrix molecules, as far as I know, and I haven't done that much research on it, to be honest, but they're ancient molecules. You know, they're conserved throughout evolution. And I'd love to know more. It's something, I've got a book actually on the matrix. Don't normally have books these days. And there is a chapter on the evolution of the extracellular matrix in living organisms, and I haven't read it yet. But we know they are quite ancient molecules. And so that's why I'm just thinking that maybe this is why it's common, you know, shared by many different cancers.
I'm going to let the right-hand side have a second go because they've been a bit slow. We'll come back.
Hello. I was just wondering, so most of the immunotherapies targeting T cells, and you alluded at macrophages in your talk, and I was just wondering if you could shed a bit of light on your thoughts around therapies that will target macrophages. Thank you.
I'm glad you asked that question, because macrophages are my favorite cells, really. I think what's going on at the moment is that there are clinical trials. The most exciting is with anti-CD47, which is a don't eat me signal, which seems to wake up macrophages. And what's interesting about that is that we've had this idea, it's a very sort of binary idea, that most cancers have got bad macrophages, that are tumor promoting, they remodel that matrix, they stimulate blood vessels, they inhibit the good T cells and all of this. But with this treatment, which has only been used in lymphoma at the moment, but with this treatment, what it seems to do is it doesn't matter if those macrophages were bad. Macrophages do lots of cleaning up in the body. They, pick up all the junk. It doesn't matter what they were doing. This switches them, and so they recognize the malignant cells. So that's probably the most promising. It's really re-educating them. The other treatments that are going on at macrophages at the moment is trying to stop macrophages come into a tumor. Now, some of our experiments in mouse models, for instance, in pancreatic cancer, were very encouraging and very, again, a model that seems to resemble the human disease. In advanced pancreatic, the anti-macrophage treatment that stopped the macrophages coming in, the T cells got woke up, and the mice survived a lot longer. But I'm not sure about that. I think that might be dangerous to take all the macrophages out. So I think the kind of treatment that's going to, if we're going to target the macrophages, is going to be, or myeloid cells in general, is more waking them up, is more activating them rather than stopping them coming into the tumor. And I think that's where a lot of research is going at this moment in time. And it's probably linked to that clinical trial I talked about. My hypothesis is that the interleukin-1 trial, the RICA trial, had something to do with macrophages. So I'm trying to get some anti-interleukin-1 beta for my mouse hospital.
And last question there.
Okay, thank you. Thinking in terms of the tumour needing the blood vessels to supply it with energy, is there a difference in terms of people with diabetes having higher blood sugar levels? Does that encourage it to grow more?
I think that's a really good question. I don't know the answer to it, but I do know that there is some evidence that drugs like metformin, which are given for diabetes, which reduce all these things, may have an anti-cancer or cancer preventative effect. And of course, we're coming into this era where there's a whole generation of people who are having statins or taking aspirin or taking drugs for diabetes. And because there is a link, like the RICA trial between cancer, heart disease, all of this, that there's little snippets of evidence that drugs like for diabetes may have cancer preventative.
And a similar thing with aspirin, I presume.
Yes. Yeah. So we're on the cusp of sort of something that could be quite interesting.
Thank you very much.
So I'm going to rather cheekily indulge and get the last question for myself, which I shouldn't, but I will anyway. Well, two questions to make it even worse. One is, I get a sense that there's almost a mechanical analogue to the things that you're showing us, that you're understanding bits and pieces moving around and how they interact with each other. Am I understanding that correctly? And the other entirely unfair question is, the trajectory of understanding of cancer and the ability to do something about it seems to be rising more steeply. The precise day doesn't matter, but roughly when do you think we'll have this cancer beaten?
Well, I'll answer that one. That's the back of the cab question. You know, you get into a black cab and he says, what you do, love? And you say, oh, I do cancer research. You found a cure yet? You know, so I'm used to that one. So my answer to that is there's 220 or so different types of cancer and everyone's, you know. So, but I'm really positive. I'm really positive. I think that a lot of cancers, and we see this already, a lot of cancers are more manageable, that people can live for longer periods of time with good quality of life. But I think that these kind of tumour microenvironment, you know, the stimulating the immune system, just attacking the support system of the cancer as well as that evolving malignant cell is another promising area. But in the end, what we've got to do is come much more into the preventative and early detection. Because if you catch a cancer before it's spread through the basement membrane, end of story. You know, so I think it's not really our research, and I think quite a lot of research will move and is moving in that way, is much more about prevention and early detection rather than, you know, curing a cancer when it's already spread. That's not a very good answer, but it's the best I can do.
Well, I think it's very encouraging, and I have to say I think particularly encouraging in your super exposition, for which many thanks, is the fact that some of those graphs, in particular cases, instead of continuing to the bottom, actually level out.
Yes, and, exactly. I mean, that's I think what excites everyone. The fact, you know, I was telling, it's a true story. You know, there's to study the end stage of cancer, there is a thing called a warm autopsy program. So when some very wonderful people, when they die, they give their bodies for immediate discovery so that people can really understand what happens at the end stage of cancer and how it evolves. And one of my colleagues in Australia had signed up a fairly young woman with melanoma for a warm autopsy, and they never got her because she had immunotherapy. And she was one of the lucky minority. And she lived, you know. They didn't get the samples, you know. You know, and you do hear stories like that. Not enough of them, but they're stories that I never dreamed I'd hear of in my lifetime. And so, you know, maybe they're just the exception. But I think, you know, there is that shoulder. There are patients who are living longer than we would have ever expected 10 years ago. But not enough of them. And that's the challenge for us.
Well, let's hope there are more, and as good citizens, we'll all sign up for the warm autopsy programme. And if the scientists get it right, we won't be called upon. Thank you very much indeed.